Welcome to solverfy.
So in this question, we're dealing with the TRIP operon, which is the operon responsible for producing the enzymes that make tryptophan in bacteria. Tryptophan is an amino acid that is essential for bacteria to obtain through their diet. And if they don't obtain it through their diet, they can make it through this series of enzymes, TRPA through TRPE. So along with all of the different enzymes or the different sections in this operon, the different genes in this operon that can make the different enzymes, you have an operator and a promoter. Okay, so those are all the different parts of this operon. Now, when tryptophan is present in the diet, you do not want to make these enzymes because you have tryptophan. So if you were to block the um, activity of this operon, then you would want the repressor to be on. Okay. On the other side, if tryptophan is absent, you want the repressor to be off. Okay. Now, if this repressor is responding to the presence of tryptophan, that means that when tryptophan is present, it binds to this repressor, and the binding of tryptophan to the repressor makes it more likely to bind to the DNA, preventing the RNA polymerase from transcribing the DNA. Whereas there's no tryptophan, the repressor is off of the operon, and then the RNA polymerase can bind and start transcribing the set of genes. So that is the overall process of how the tryptophan operon is controlled. Now let's look at our answer choices. RNA polymerase binds to the repressor. RNA polymerases don't bind to repressors. They are recruited by transcription factors that bind to the promoter. Choice B, the repressor binds to the promoter. No, because as we just established, when you don't have tryptophan, you want the repressor to be off. If the repressor is on, there's a mutation. Choice C, the repressor dissociates from the operator. Yes, that is absolutely correct. That's what's happening here. So choice C is correct. Choice D, RNA polymerase dissociates from the promoter. No, it does not. The repressor doesn't affect how the RNA is being recruited to the promoter by transcription factors. It only prevents the RNA from being from moving forward and transcribing.